Hey guys, we're moving on to PowerPoint 2 today, and we're moving on to DNA replication. So Unit 4, Topic 1, PowerPoint 2. So this is Pearson Biology 12, Section 6.1. And by the end of the lesson, you need to be able to explain the role of helicase in terms of unwinding the double helix and separation of the strands, and DNA polymerase in terms of formation of new complementary strands in the process of DNA replication. Reference should be made to the direction of replication. Now, the basics of cell replication is that cells have to copy themselves, and that's a normal part of a cell's functioning. The cell theory states that all cells arise from pre-existing cells. So for this to happen, cells need to be able to replicate and copy everything inside the cell. And this is mitosis. We would have learnt this in year 10, and this happens when our cells need to grow, divide, replenish damaged cells. And as we can see in the figure, our DNA starts as a singular copy of all of our chromosomes, and they double themselves. We end up with a double set of all of our chromosomes. In the process of mitosis, these chromosomes line up at the center of the cell, and we have this special machinery come in and grab onto the chromosomes. As the cell divides, it's going to pull an equal copy of each chromosome to one side of the newly forming cells until we get two new cells that are an identical copy of what we started with. That's happening all the time, especially when we're going through periods of growth. Areas like our stomach, which are replenishing all of the time, this is happening constantly. Now, this is giving you an overview of largest to smallest. Remember, I mentioned in the previous PowerPoint that it's good to orientate yourselves with what we're actually talking about. Yesterday, we looked at DNA structure, which was looking at the double helix and the different components of the DNA. Now, remember we talked about DNA has to package up. It wraps around those histone proteins and packages into these really tight coils to fit inside the chromosomes. And we have 23 pairs of chromosomes in the nucleus of each one of our cells. So every individual cell in our body has all of these copies of our genome packaged inside these chromosomes. The only exception for that is the red blood cells, which don't contain any DNA, and our sex cells are a little bit different as well, which we'll get to. And a huge collection of these cells is what goes into making an organism. So you can take a skin swab and you can see all of your chromosomal makeup inside one of those individual skin cells. Now, DNA replication is the name of the process of the copying of our genetic information. It's really easy to look at mitosis and go, oh, yep, everything copies and it splits in half. But we're going to look at the process by which the DNA itself actually copies. We need the DNA to do a copying and pasting of itself, and it's quite a complex process how it does that. We have approximately 3 billion base pairs in our genome, and all of that has to copy every time one of our cells divides, which is happening pretty constantly. This image on the right shows an overview of how this works. So we go from our double-stranded DNA helix, and you can see that the double strands begin to unwind from each other. Now in the orange here, you can see where a new double helix is starting to form off each of those individual strands. And this continues until we have two identical copies at the end here. And that's the process of DNA replication. It's a copying and pasting of our DNA. Now, one point of confusion when we start looking at cell division is looking at the different states that chromosomes exist in. And we're really used to seeing chromosomes drawn in this fashion in the middle here, which is actually how chromosomes exist after they've been copied. In the cell, normally, we have chromosomes existing of one singular chromatid, which is basically like a half of a chromosome. When they are going to copy in replication, they duplicate to form this X structure that we commonly associate with chromosomes. But actually, we refer to this as sister chromatids. They're two chromatids that contain the same information. In mitosis, we then split those two sister chromatids apart so that we have an identical copy of the daughter chromosomes, one going into each of the new cells. Now, the cell actually exists in a cell cycle where this is sort of a representation of the life cycle of one particular cell before it divides. We can see on the image on the right that a large proportion of the cell's time is spent in this phase called interphase. And this happens before cell division. So majority of the cell's time is spent in this phase where it is basically copying all of its material. It copies the DNA and it copies all of the components of the cell and gets ready for division. This green part is mitosis. This is actually the splitting of the cell, which we can group into prophase, metaphase, anaphase, and telophase. This very last phase in the blue is called cytokinesis, and this is the part where the two cells actually butt off from each other to make two new daughter cells. Now we're going to look at mitosis in a separate lesson here. The focus of today's lesson is on the DNA replication, which is happening specifically in interphase in this portion here called the S phase. We'll look at this in later PowerPoints, but S phase is talking about synthesis, copying of DNA. We won't talk about the cell cycle now until we look at cell division, but today we're going to continue with thinking about how are we copying the DNA, what's actually happening in this S phase in order for DNA to copy. This is an overview of mitosis, which, as I said, we'll look at in separate lessons, but this is just to give you some idea of what's happening after the DNA has copied. Now, when we look at prophase, we already see these X-shaped chromosomes, and that's because of what has happened in interphase. The cell doesn't normally exist in this chromosomal form. Remember, I mentioned earlier that it exists, like we see over to the right here, as singular chromatids. Interphase's job is to copy these chromatids into structures like what we have here with prophase, because they're going to be split in the process of mitosis. A general overview of this just before we proceed. Our chromosomes condense inside the nucleus, and we actually see the membrane of the nucleus start to break down. The chromosomes leave the nucleus. There is no longer a nucleus for that period of time because the membrane's broken down and the chromosomes are in the center of the cell. We have this special apparatus called the microtubule, which we'll talk about later. And this comes along and joins up to the chromosomes and grabs them. And eventually we're going to be pulling these different chromatids to the separate sides of the cell. In telophase, we're completely moved to the ends of the cell. And this phase here is this cytokinesis phase, where the two new cells split from each other and we have two identical cells. Think about this when our skin cells are replenishing themselves, or when our stomach lining needs to replenish itself. This is how this works. And then this daughter cell will go off and undergo interphase again and mitosis when it's ready to divide. Remember, today we're looking at how that interphase bit happens. How does it copy? So we're not going to focus on the steps of mitosis here. That's for a later date. What we're looking at today is how the DNA replicates itself in this process. If we have a look at the image on the right, we've got our DNA double helix, and we know that our base pairs in between are the coding aspects of the DNA. The general gist of DNA replication is that our two strands of our DNA double helix get split apart from each other, and each individual strand acts as a template for the production of a new complementary strand. So see in this image how the blue strand is the existing blue strand that we had before, as well as this kind of aquary teal color strand on this side. They're the existing strands from our DNA molecule. The green strand that is getting built off of this is our newly forming strand, making complementary strands off the existing template strands. We call this model a semi-conservative model because each newly formed double helix at the end here contains one original strand and one new strand that's been synthesized off the template strand. Now, this happens in the 5' to 3' direction. So remember yesterday we spoke about those carbons. We have to think about the direction that this is happening. And I'll refer to this again a little bit later. Remember, there is a video that you can watch that goes through the direction of a DNA molecule. Now, this is an image from your Pearson textbook where we've got the parent DNA molecule here on the left. So this would be what's inside our individual chromatid. Now, each one of these beigey sort of taupey colored strands is going to be preserved. And from that, we're going to get a newly synthesized complementary strand shown in blue. Now, both of these both of these identical double helix strands are an exact copy of what we started with the parent DNA. We've just copied and pasted them. Now we're going to go through the steps involved in DNA replication. And a lot of my resources and images in these slides have come from Khan Academy, a wonderful resource that you can use and really good visualizations of this process. Now in the syllabus, it mentions that you need to know the role of helicase. And helicase is an enzyme which unzips the double helix. So the helic part referring to the helix, A's referring to it being an enzyme. Now, if we look at the image here on the left, helicase is going to unzip the strands and it creates this parting in the strands, which we refer to as a replication fork. If we look at the image from Khan Academy on the right, we can see that the hydrogen bonds that are holding our base pairs together here are now broken, and we've parted those bonds to separate two different strands. The syllabus also mentioned DNA polymerase, another enzyme that's really important in adding DNA bases. Later on, we'll talk about RNA polymerase, which adds RNA bases. DNA polymerase refers to adding polymers, so making a chain of DNA, A's again referring to it being an enzyme. So DNA polymerase moves along our template strand of DNA and it adds complementary bases to our template strand. Each of those two strands from the original double helix acts as a template, and a new strand is being synthesized as we add bases to our existing strand. In terms of the direction, we can see that this new strand is being built in the 5' to 3' direction. So it's moving along the template strand in the 3' to 5' direction. Another really great figure from Khan Academy here is showing this in a visual representation. So here we've got our old DNA strand running in the 3' to 5' direction. And we have our DNA polymerase, this large enzyme that's binded to our strand. And it's adding DNA bases going this way in the 5' to 3' direction, chugging along the template strand and producing a new DNA strand. You can see in this image here, it's adding the bases as it goes, moving along in that particular direction. At the end of replication, we end up with two identical DNA double helices. Each has one new strand and one old strand from the existing double helix molecule. There's a video here that you can watch in my PowerPoint if you click on the link. So from this lesson, you need to be able to explain the role of helicase as well as DNA polymerase. You need to talk about the process of DNA replication and particularly how those enzymes do the job. It also says that you need to make reference to that direction of replication. So it's important that you look back and understand that 5' to 3' idea. The syllabus specifically states that you don't need to know the different DNA polymerase enzymes. There's actually 2 different ones called 1 and 2. You don't need to know that level of depth, so if you come across it, don't stress about that. Now, there are some questions in your Pearson textbook, and I'm also going to upload a blank copy of the BioZone handout from the DNA and Inheritance book that you can complete in the OneNote.